Today we are talking about the origin and development of the central nervous system. The central nervous system appears at the beginning of the third week of embryonic development in front of the primitive node. This area here starts to thicken to form the neural plate. Then the lateral edges of the plate will start to rise above the embryonic ectoderm to form the neural folds. With further development, the neural folds continue to elevate and approach each other, then to fuse at the future cervical region of the embryo to form the neural tube. This fusion proceeds in cephalic and in caudal directions to create a hollow structure that is lined with the neuroectoderm epithelium. The cranial and the caudal part of the neural tube at this stage are still in open communication with the amniotic cavity. The cranial neuropore closure happens at day 25th of development, whereas the caudal neuropore closure occurs at 3 days later. By day 28th of development, the cephalic portion of the neural tube shows 3 dilatations, what's called the primary brain vesicles. The first one is the prosencephalon, or the forebrain. Second one is the mesencephalon, or the midbrain. The rhombencephalon, or the hindbrain. At the same time, 2 flexures form. The first one is the cervical flexure that marks the boundary between the rhombencephalon and the spinal cord. The other one is the cephalic flexure, which is located at the midbrain region. By 5th week of embryonic development, the 3 brain vesicles have differentiated into 5 secondary vesicles. In this sense, the prosencephalon will form the telencephalon and diencephalon. The mesencephalon stays the same. The rhombencephalon will form the metencephalon and the myelencephalon. At the same time, the rhombencephalic isthmus will separate between the mesencephalon and the metencephalon, whereas the pontine flexure will mark the boundary between the metencephalon and the myelencephalon. The derivative of the telencephalon will give to the cerebral hemispheres. Diencephalon will form the optic vesicle, thalamus, hypothalamus, and pituitary gland. The mesencephalon will form the anterior visual and posterior colliculi, auditory colliculi. The metencephalon will form the cerebellum and pons, whereas the myelencephalon forms the medulla oblongata. The lumen of the spinal cord, the central canal, is in open communication with the cavities within the 5 secondary vesicles, brain vesicles. Thus, the cavity within the rhombencephalon will form the 4th ventricle. The cavity within the diencephalon will form the 3rd ventricle. And the cavity within the cerebral hemispheres will form the lateral ventricles. So once the neurotube is completely closed, the neuroepithelial cells begin to give rise to another cell type, the primitive nerve cells, or the neuroblast cells. They form the mantle layer at a zone around the neuroepithelial cells. The mantle layer forms the gray matter of the spinal cord. The outermost layer of the spinal cord, it's called the marginal layer, contains nerve fibers emerging from the neuroblast of the mantle layer. As a result of the myelination of the nerve fibers in this region, it takes a white appearance, and therefore it's called the white matter of the spinal cord. And as a result of the continuous addition of a neuroblast to the mantle layer, each side of the neural tube shows a ventral and dorsal thickening. The ventral thickening, the basal plates, contain ventral motor horn cells. It forms the motor areas of the spinal cord, and the dorsal thickening, the alar plate, contains the sensory areas. A longitudinal groove, the sulcus limitans, marks the boundary between the two thickening. The dorsal and ventral midline portion of the tube, known as the roof and the floor plates, they do not contain any neuroblasts. They serve primarily as a pathway for nerve fibers crossing from one side to the other side. In addition to the ventral motor horn and the dorsal sensory horn, a group of neurons accumulate between the two areas and form a small intermediate horn. This horn is containing the neurons of the sympathetic portion of the autonomous nervous system and is present only at the thoracic T1 to T12 and upper lumbar levels L2 to L3 of the spinal cord.